In this lesson, we are going to learn some basic things about viruses. As you probably are familiar with the word, viruses cause cold and a lot of other illnesses. So, what are viruses? So, viruses are defined as infective agents that are acellular, meaning they are not composed of cells. Remember, cell is the term used to describe structures that makes up living organisms. So this should tell you that viruses are not considered alive. They're not considered living organisms. They typically consist of some form of genetic material, which could be either the DNA or the RNA, but they never have both. A protein coat or capsid to protect their genetic information, and some of them they have a lipid envelope sometime present around the protein coat when the virus is outside of the cell. Now this lipid envelope is acquired when viruses leave their host cell. They take a part of the host cell membrane and convert that into their envelope. Why viruses are not considered alive? Now, one of the properties of life is to be able to carry out metabolism. Metabolism requires enzymes. Enzymes are proteins synthesized by ribosomes. Now, since viruses don't have any type of cellular structures, they lack ribosomes as well. Since they don't have ribosomes, they can't make protein. Remember, proteins are essential for metabolism. This makes viruses totally dependent on their host to synthesize their molecules and also to reproduce themselves. They are the only type of microorganism studied under microbiology that cannot reproduce without a living host cell. The viruses come in various forms. We'll look at that in next slide. They could also be enveloped or non-enveloped. If they are enveloped, we call them enveloped viruses. They cover themselves with a modified section of a cell membrane, creating a protective lipid envelope. Remember, they acquire that while leaving the host cell. Some examples of enveloped viruses are influenza and HIV virus. This image is showing you three different forms of viruses: helical, polyhedral, complex. How do viruses multiply? Since they're not alive, they don't have any metabolism, so how do they reproduce themselves? So this is how it works. After contacting a host cell, the virus inserts its genetic material into the host cell, takes over that host cell's function. The entire metabolic machinery of the host cell now only serves the virus. After infecting the cell, the virus continues to reproduce, but it reproduces more viral protein and genetic material instead of the usual cellular products. Again, the host cell doesn't make anything for itself. Everything is made for virus only. It is this process that earns viruses the classification of parasites, or rather an obligate intracellular parasites. Intracellular parasites because they can only multiply once they're present inside their living host cell. There are two types of multiplication methods used by viruses. The first one is called lytic cycle, which always ends in the death of the host cell right away. The second one is called lysogenic cycle. In this case, the virus lives inside the host cell by integrating its genome into the host genome and becomes part of it for various generations. Then at some point, The viral genome that has been integrated into the host cell can splice itself out of the host genome and then go into lytic cycle, making copies of the virus particles which are released by death of the host cell. Image is comparing both of these life cycles. So let's please follow the images. So here is the lytic cycle. Lytic word comes from lysis, which if you remember means breakdown. The phage infects the host, injects its DNA into the host. The minute that happens, the viral genome takes control of the host cell. The host cell DNA becomes inactive. It cannot do anything for the host cell, but everything is done for this infective agent, the virus. The virus genome is multiplied. The genome is used to produce viral structural proteins. It's a stage that is called biosynthesis. And look, once everything is synthesized, the virus particles are assembled. And if you look back in the initial first stage, this is a complete virus particle with a copy of the genome. And that's what's going on here inside the cell. Once all these copies are produced, the host cell dies. The lysis of the host cell wall releases these viral particles into the environment, which then can go and cause infection of the new healthy host cell and keep carrying this cycle over and over again to multiply themselves. Lytic cycle. Some of the viruses that are referred to as temperate viruses, temperate, meaning they're not very aggressive. Once they inject their DNA into the host cell, the DNA of the virus becomes part of the host genome. Look at that. The red molecule of the viral genome is incorporated into the blue genome of the host cell, in this case a E. coli, a bacterial cell. When the bacterial cell multiplies, it replicates its DNA, it replicates the DNA of the virus along with it. And that viral genome is passed on to the next generations of the bacteria. As you can see, this is a bacteria undergoing binary fission. Now, each one of the daughter cells then goes on to grow, become a fully formed mature cell, and this cycle can continue for various generations to come. At some point, nobody really understands, we don't understand that yet, but something triggers and the viral genome can splice itself out of the host genome and can enter into the lytic cycle, leading to the reproduction of the viral particle known as virions and ending up at the death and the destruction of the host cell. So in this lesson, you were introduced to some of the basic properties of what viruses are, why they're not considered living, why do we call them obligate intracellular pathogens, and the distinction between two ways they multiply themselves, lytic versus lysogenic.